Assalamualaikum warahmatullahi wabarakatuh and good day. I am Madam Ros Azinda with Teacher Ramin. Today we are going to continue our lecture on cell membrane. Other function for the cell membrane is for the biotransport. Given the selective permeability of lipid bilayer, a number of mechanisms exist by which substances are moved across lipid. Which movement across membrane is important, for instance, as a means of removing waste from a cell or bringing food into a cell. Okay, this is the example for the biotransport. Here, the translocation of the solute from one side of a biological barrier to another. For example, here, from the phase 1, in the intestinal content, okay, to the phase 2 in the bloodstream, water and glucose can be transported from the intestinal area to the bloodstream. Okay, for the cell transport, plasma membrane regulate traffic in and out of the cell. Since it is selectively permeable, some substance may pass freely and some may be needs a specific transporter and some may be prohibited. There are three ways of the cells transport. First is a passive transport, which is substance move from higher to lower concentration, which is no additional energy required. Second is the active transport, which substances move from the lower to higher concentration, which is additional energy is required. Lastly is the bulk transport, where the movement independent of the gradients and also additional energy is required. Okay, here is the illustration which are how the substance can pass through the cell membrane. Maybe water and other non-chargeable molecule can easily pass through the cell membrane, while the charged molecule and ion would need some special transporter or channel to pass through the membrane, and some big molecule cannot pass through the cell membrane. Okay, for type of passive transport, first is a diffusion. A solution consists of a solvent and also a solute. Diffusion is a net movement of solute molecules from where there is more of is along a concentration gradient to where there is a less of it until molecules are equally distributed. In terms of cellular activity, diffusions do not require energy. However, the cell has no control over the diffusion and the rate of diffusion is quite slow. Okay, this is the example of the illustration of the simple diffusion. Okay, here is a water added with the red dye. So if given sometimes, okay, the molecule, the red dye can move, okay, from this region, okay, across the membrane freely. At the equilibrium, the dye equal on both sides of the membrane and net movement stop. So the solution contain the dye and others contain the dye and also the solvent equally. Factors affecting diffusion rate could be steepness of concentration gradient, which is steeper the gradient, faster the diffusion. Molecular size, smaller molecules, faster the diffusion rate. temperature. Higher the temperature, the faster diffusion rate, electrical or any other pressure of the gradients. Now we move to the osmosis. Osmosis is the net movement of water, diffusion of water through a selectively permeable membrane from a region of greater water concentration, which is a low solute, to a region of lower water concentration, or which is a high solute. It is a special form of diffusion. The solution have an osmotic pressure due to the presence of the solute and often involve movement of the water. Here is the illustration for the osmosis. Diffusion of water molecule across the selectively permeable membrane. Direction of net flow is determined by water concentration gradient. So site with the most solute molecule has lower concentration. So here, concentration of water is very high and the concentration of water here is very low. So the concentration of water from this region will move into this region until it reaches the equilibrium. Okay, this is another example of the osmosis demonstration. You can read this later. Okay, now we move to the effect of the osmosis on the cells. Okay, for isotonic solution, okay, cell neither gain or loses water. This is because the concentration of water same on the both sides of the membrane, mean that the solution and the red cell itself have a same pressure of the gradient. So 0.9% of saline isotonic to the red blood cells. So here is a hypotonic solution. Concentration of water outside the cell is greater than inside the cell. This means the water is more outside the cell. So it causes the cells to gain more water. An animal cell may lyse or burst, and while the plant cell used to remain or to get due to the cell wall. Okay, lastly, in the for the hypertonic solution, the concentration of water outside the cell less than inside the cell. So this will make the cell lose of water. So the water move from the red cell outside to from the outside from the red cell. So the animal cell will shrink while the plant cell undergo the plasmolysis and may wilt. Okay, now we move to the facilitated transport, which is involve the carrier protein. So what happened here for facilitated transport, especially for the small molecules like glucose and amino acid, they can't get through the membrane lipid. So it needs a special protein to combine, which is most of the time what we call as a carrier protein. So it can be moved from the one spot to another, from outside the cell into the cell, which is do not require any ATP. So this is another process of the facilitated diffusion, which the protein can bind with the molecule and the shape of the protein changes and molecule can move across the membrane. Let's say from the outside, the cell goes inside the cell. Okay, for the active transport, okay, cell expend energy to move molecule against a concentration gradient. It requires a transport protein, and most of the time it involves the sodium potassium pump, which is important in maintaining the gradient of iron used in the nerve impulse conduction. Okay, for the bulk transport, macromolecules are often too large to be moved by the transport protein. So the vesicle formation takes them into in or out of cell. There are two types of bulk transport. First is the exocytosis, which is the movement out of the cells. Here, from the plasma from the cell inside the plasma membrane. So exocytosis happen. Okay, bring out the molecules or whatever particles are outside to the membrane. While the endocytosis movement into the cells, which can be divided into three: phagocytosis, pinocytosis, and also receptor-mediated endocytosis. For the phagocytosis, cells surrounds and engulf and digest the particle, while the pinocytosis, vesicle form around the liquid or small particle, and the receptor-mediated endocytosis, which receptor for particular substance found in the coated pit and selective and more efficient. Here it is the endocytosis. From the outside the cell, move inside the cell. Okay, this is the phagocytosis. Phagocytosis, this is pinocytosis. Okay, for detail of the phagocytosis, how it happened. Membrane surround and engulf the object used for larger object and pinch off, placing the particles in the phagocytic vacuole. The phagocytic vacuole then fuses with lysosome and the material is degraded. For example, for a unicellular organism like a amoeba and also white blood cell for macrophage. Okay, for method of the pinocytosis, the process by which the cell takes in a extracellular fluid or very small particle by the invagination of the cell membrane. A pocket then form and pinch off to form a vesicle. The liquid content of the vesicle is then slowly transferred to the cytosol. For example, it happens in the blood cell, also in the plant root cell. Okay, lastly, for the receptor-mediated endocytosis, a form of pinocytosis occurs when a specific macromolecule such as vitamin, lipoprotein bind to a plasma membrane receptor. The macromolecules are taken into a cell via coated vesicle that pinch from the plasma membrane. Molecules first bind to specific receptor protein which are found at one location in the plasma membrane. This location is a coated pit with a layer of fibrous protein on the cytoplasmic side. When the vesicle is uncoated, it may fuse with a lysosome. So this is how the illustration of the receptor-mediated endocytosis. So this is the receptor protein bind with the specific molecule. When this is become a coated pit and then it will engulf, it become pinch and become a coated vesicle. So this is the same illustration. Alright, end of the lecture. Thank you very much for your attention.